once again, I, Ajay Jha, on behalf of ACS India team, welcome you to today's ACS Science Talks. Just to update you on the upcoming ACS Science Talks, please visit our ACS in India webpage and register for the upcoming sessions. We'll post the details on the new sessions soon. We have also uploaded the past lecture recordings on the ACS Science Talk virtual library section. Please feel free to go live, subscribe, and share among the scientific community. To update you on the latest research at ACS, the products and launches, we have monthly newsletter ACS Insights India. Subscribe today for free. We'll provide the subscription link into chat box later. Now to begin with the scientific session, it's a pleasure and honor to have Professor Jonathan Sweedler with us. Professor Sweedler received his PhD degree in chemistry from the University of Arizona in 1988 and spent 3 years at Stanford before moving to the University of Illinois at Urbana-Champaign in 1991. He is currently the director of the School of Chemical Science and James R. Eisner Family Endowed Professor of Chemistry, as well as a Center for Advanced Study Professor of Chemistry. He is also affiliated with the Institute of Genomic Biology and the Beckman Institute for Advanced Science and Technology. Before being appointed as editor-in-chief of Analytical Chemistry, he served as an associate editor for 4 years at Analytical Chemistry. Professor Sweedler has established an international reputation for designing innovative analytical methods. He has created novel metabolomic and peptidomic technologies that can be used to discover the roles that peptide hormones, neurotransmitters, and neuromodulatory agents play in behavior, learning, and memory, a glimpse of which he'll share with us today. He has received several recognitions, including the Malcolm A. Pruitt Award by Council for Chemical Research, Torbjörn Bergman Medal from the Swedish Chemical Society, Anachem Award by the Federation of Analytical and Spectroscopic Society, Fellow of the American Chemical Society, Victor Mudd Prize by International Regulatory Peptide Society, and many more. Now, I would like to invite Professor Sweedler to deliver his talk.
Thank you very much for that wonderful introduction. And okay, so today's talk, which is typical, we'll start out with just a few minute introduction to the Journal of Analytical Chemistry. And then I'll talk about some research, and then I'm happy to answer any questions. I'm delighted to be here on this Friday morning in Illinois. If you don't know, Champaign-Urbana is about 150 kilometers south of Chicago. So sort of in the countryside, surrounded by corn and soybeans, a little bit different than where most of you are. So Analytical Chemistry is one of the older journals of the American Chemical Society. It started actually in 1929, and the editors, especially the first few, were editors for quite a while. So I've been an editor for, this is my 10th year, and I've had a lot of fun doing it. Analytical Chemistry publishes a couple, it says 2,000 articles in 2019, and about a third of those are from the United States and 2/3 from the rest of the world, amazingly evenly distributed between the Americas, Europe, and Asia. And so that's one thing that's been fun to look at. It's the highest impact journal in analytical chemistry, and it does quite well in terms of citations. I also like, we just started a new cover series that's sort of aimed at highlighting diversity in the chemical sciences. And we have people submitting art, which is a little bit different than we've ever done before, because it's not just, so one issue a month now has a cover that's not related to an article. And here's just one of them shown here. And that's been a fun program. So I just thought I would put this up. There's a lot of ways of looking beyond impact factor. And in most of them, analytical chemistry does well. Google Scholar actually does H-index for journals. I was sort of surprised about that. Most people know what an H-index is, the number of, if it's a person, it's the number of articles that received at least that many citations. This is saying, our H5 index is quite good. And lastly, maybe not surprisingly, in terms of being cited by patent literature, this is something that is tracked. We're, in, I think, depending on which year, 5th, 6th, 7th journal, higher than all the other, for example, ACS journals. That's not surprising, maybe, for a journal that emphasizes new technologies. It means the patent literature cites us quite a bit. So let me just go over a couple slides about the journal. It's analytical chemistry, it's chemical measurements. And I can tell you it's worth looking at the scope, because as an editor, one of the most, I would say, frustrating things is when you get good papers that just don't fit the journal. You may have the world's best synthetic chemistry paper. You submit it to us, it won't be published, and that's obvious. But there is other things to be aware of. So we do publish fundamental or applied, and any phase of the analytical operations with some of the topics listed here: chemical reactions, chemometrics, electrochemistry, goes on to instrumentation and others. It's an evolving list as instruments evolve. But the important point here, and I see that some people misunderstand this, if you're using a method that's already been established, for example, I'll pick one that I use a lot: liquid chromatography, mass spectrometry. And yes, that's an analytical method. And you have a new type of sample, whether it's you're going to look for pollutants, a different pollutant in a river water, or something on human skin. And your graduate student spends a year or so trying to optimize that, but you're using really what is standard LCMS, but you've just done an optimization of the sample prep. That type of article we tend not to publish. What we publish is articles that advance the techniques or have not been done. And so as another example, if you come up with a new fluorescence probe, and I will mention this specifically for glutathione, and it's a new probe that's never been done, it's a fluorescence probe. There's already actually about 200 or 300 glutathione fluorescence probes already measured. Why do we need another one? What about your new probe enhances the measurement, enables a measurement to be done that has never been done before? And so just because the probe is new doesn't mean we're interested if it's a common measurement. And this can include, glucose, it could be glutathione. I mentioned the glutathione because in the last week, I think we had 11 articles submitted on glutathione fluorescence probes. They're all unique, they're all different, but none of the articles explained what they enabled that the previous hundreds did not. And so again, for a chemical measurement journal, we're interested in what is enabled by your technique, what hasn't been done before. So it's not just how well your approach works, but how well does it work within the context of the field? And so that's an important point. And I'm not going to go into this. This is the same for most journals, but I wanted to put this up for two reasons. You submit your article, it comes to the editorial team, and we look at it. And if it doesn't fit the scope, or I don't see, for example, in a case of a glutathione probe, what makes it better than any of the others in the past, or then our editorial team, oftentimes consulting with another associate editor, will make a decision before sending it out for peer review. I often get comments that this isn't fair. But to be honest, the other way to look at it is if it's scope, for example, I don't really want a reviewer's comment. If it doesn't fit our journal and fits, for example, the Journal of Proteome Research or Environmental Science and Technology, that is actually an editorial decision, not really a reviewer's decision. So this is how it goes. And the reason I mentioned that is I was looking at papers from India, and I noticed very few have detailed cover letters. Some do. And the cover letter that I get that is sort of frustrating from an editor's point of view, it tells me that here's my article. It's not been published elsewhere. I know I do the same thing. You spend a long time on a paper, and as soon as you're ready to submit, you want to submit. You don't even want to think about a cover letter. A cover letter is the chance for you to sit over my shoulder and tell me about your paper. Tell me why it's important, why it fits the journal, especially if it's not obvious, and what the take-home message. The last point I'll make about this is that I find it frustrating as I reject a paper because of scope or something, and then the authors come back and write an appeal, and they have a really good reason why the paper fits analytical chemistry. And what I find frustrating about that should have been in the cover letter. So I encourage you, as you submit papers to my journal, Analytical Chemistry, but any other, spend a little thought on the cover letter, because it's the time you get to tell the editor. You're sitting behind his shoulder saying, hey, this is why my work is good. So I'm just going to end the journal with just a couple quick things. Besides research manuscripts, we have features and perspectives and reviews. Different editors or associate editors handle these. And here's some of the information about those. If you're interested, let us know. Reviews, please contact us in advance. We only have one issue a year, and we actually won't publish two similar reviews on the same issue. So you might have a great review, but we've already have one from somebody else that's not going to come in. And so please contact us in advance. Here's the team. And this is just a part. I didn't want to show 20 associate editors, on a picture, but just showing you it's a very international group. And then Going one step further, we do have two editorial advisory board members from India currently, and this is an evolving, so the EAB evolves. Everybody serves 1 3-year term. Some people get like, wait, what did I do? Why am I off? And the whole idea is we can add new ones. And so we're always looking for people willing to lend their name and help promote the journal. And we try to also promote the individuals. Here is just a few highlights from some articles I just picked. This is just one of sort of each type published in the last few years in terms of 1,000+ downloads in just a couple of years. On this article here, we have a review that was published basically this year, January 1st, the first issue, and then a perspective and a feature. And so they all do well, and I encourage you to send your best chemical measurement science to analytical chemistry. And then one last take-home message before I start my science talk, and that is we do give away. We award 2 different awards. One of them is the Advances in Measurement Science Lectureship Awards. These we pick 3 winners each year, and that's from the Americas, Europe, and then Asia Pacific. And then normally they have traveled to a meeting each year, and the meeting is now it used to be PitCon, now it's going to vary. This year it was virtual. I'm not really quite sure what's happening next year, but it will eventually be a trip to a meeting. One of the fun things about this award is it's on recent major impact, think 5 years. So you can be an assistant professor starting out and you've made a really good contribution. You can be a senior person who made a really good contribution in the last 3 to 5 years. And that's the basis of selection. So you could have made a great contribution 30 years ago, and that's great for you, but it doesn't really fit this. This is supposed to be something fairly recent. The only other thing I'll say is that this is not an analytical chemistry award, but this is combining with the other measurement science journals of the American Chemical Society. And so that would be ACS Sensors, it would be Journal of Proteome Research, and now JASMUS and a few others. And this is really a fun thing. And these are the award winners, or the nominations are open for this coming year. And truly the last general slide is ACS is trying something called a forum. In this particular case, it's an inaugural measurement science symposia. It's coming up in June, and all the journals are have different sessions. The session that Analytical Chemistry is sponsoring is New Tools to Study Viruses. We have some great speakers, and then there's it goes for the whole week. And these other journals also have really fun symposia. And so this is something, if you like the idea of these types of talks, this is a virtual event that'll be occurring actually, I guess, in about 3 weeks. So that's it for the journal. I'm happy to answer any questions. And I will also add that I'm happy to answer any questions via email if you don't get them answered about what the journal is like and what we're looking for. And so, don't hesitate to contact us. So now let's make sure. Yeah, okay. So now today I'm going to talk to you a little bit about some of the measurement sciences approaches we're using in my group to study the brain. This is actually a statue, right? That I'm seeing, if you're wondering what this is this is just one of the icons of the University of Illinois. But today what we're going to do is talk about ways to study the cells of the brain. Now, I tend to end with an acknowledgement slide, and I never have any time to go over it. So I thought I would start with an acknowledgement slide, and this just shows the group over the last 5 or so years, and some of the work the people that did the work. Obviously, the most recent picture is a typical Zoom meeting from last month. This is more in the lab. And then Illinois can get cold. It can be -20 centigrade in the winter. And it can be 40 centigrade in the summer. So it has a varied weather. But these are the people who have done the work. So this is a chemistry talk. And so it may be a little bit funny to start with 2 slides that talk about the brain. But there's a lot of questions about how the brain works that really have remained intractable. Sort of a silly question to start with is, what is consciousness, or even what is memory? And that has been studied for 2,000 years. It's really exciting to me that we're actually at the point now we can answer parts of that, and we may actually understand this, in the next 10 years to the point where we can really understand and decipher, for example, read out what a memory is in a simple organism like a fruit fly. The reason I started with this is that a lot of the advances in understanding how the brain works really came about because of new tools. And what I'm going to do today is focus on tools that allow us to go to the cell scale. The brain is different than a lot of other organs because memory function really is cell specific. This is just showing a really pretty picture of individual neurons and the connections between them, and then lots of empty space. The empty space is astrocytes, oligodendrocytes, and other cells. If you understand this type of complexity, you really have to go to the cell scale. Otherwise, you really don't get the right answers because these cells are different from each other, and those are what we're after. So what are the molecules of a cell? And what are the molecules of a brain? Well, most people think about, DNA and RNA. And if you actually go to the smaller molecules, lipid maps, an international consortium looking at lipids, really has a list and says there's about 50,000 different chemical entities that are the lipids of the brain. And these change very drastically and dramatically during development, during learning and memory and other processes. And then there may be 50,000 metabolites. I put that down because, to be honest, most of the molecules in the brain are lipids and metabolites. They're not proteins and DNA. And so that's what I'll be concentrating today on. And again, we want to be able to measure this type of complexity at the single cell level. And we're not there yet, but we can measure some of this complexity, and that's what I'll be showing you. Single cell measurements aren't new. It started, you could argue this if you want, but I like to think that single cell measurement started with the development of microscopes, maybe by Leeuwenhoek or Hoek, depending on which year you want to look at. And they got to name the cells. Within the brain, microscopists name the cells and they pick shapes. They're star-shaped, stellate, pyramidal cells, and others. If chemists had named cells, which we started to do in the 60s and 70s, they would have named them for things like they're serotonergic or dopaminergic. They use dopamine or other important molecules. But unfortunately, the shape comes first. And so that's why most of the neurons have names related to shape. And then lastly, the last 30 years have sort of been wholesale measurements. And that's what I'm going to focus on today. I'm not going to go into the techniques too much, but my group is a mass spectrometry group. We use expensive tools. This is listing a few. This is more of a laboratory constructed SIMS, secondary ion mass spectrometry device. And then these are some commercial devices we use. They happen to be Bruker devices because we're a Bruker Center of Excellence. Other instruments would work well also. And so let's ignore that part and I'll just simply sort of talk about the measurements that we can do with them. So today's talk is on single cell measurements. And one way to do this is if you have steady hands and you don't drink too much coffee, you can actually, isolate a cell, put it on a target. And then as shown here, if you add a matrix and you blast the cell, it's kind of a silly looking cell, with a laser, you blow it up. It's almost Star Wars-like, and you see particles going up. Those are the ions coming from the cell, and you can detect those with the mass spectrometer. And shown here is a mass spectra where each peak is a peptide found in that cell. Well, actually, this cell right here. So basically, that's the technique of matrix-assisted laser desorption ionization mass spectrometry. The reason I show this slide is twofold. If you hit this cell, you'll see what's there, and if you get another cell off this picture, you'll see another. And the other is after you're done, a lot of the cell is left and you can measure it again with another technique. And that makes this approach fairly unique. Why do we do this? Well, my group, we might want to understand learning and memory in a person. My group works on animals ranging from mammals. We've done mice and rats and even camels and things all the way down to corals. So this is in between. I'll show you examples. This is a sea slug that can learn what to eat or not what to eat. Here's a part of its brain. Each of these are cells with, you can see the processes on top using EM, and this cell is missing. This cell is missing because we pulled it out and made this spectra. And these are some peptides from a biochemical point of view. It's phenylalanine, methionine, arginine, phenylalanine, amide, or formurfamide. Only biochemists would learn how to say the amino acid code like that. But this peptide and some of the others found in the cell help drive, when this cell is excited, into swimming behavior. So we can link Even a chemist can say, well, if we discover peptides in the cell and we know what the cell does, maybe those molecules have something to do with function. And so this approach where we do staining to find the cells, we remove the cell, and we do mass spectrometry has allowed my group to discover not just literally thousands of brain peptides in lots of different animals, but in about 100 cases to figure out what they do. And that's one advantage of single cell measurements. The problem with this approach is it's slow. Take somebody isolating cell by cell. So today I thought I would show you how to make this a lot faster. We can take the cells and scatter them on a target. There's the cells, there's other pieces of processes we don't have to worry about. If we shine our laser, not with mass spec imaging and form a raster pattern, but if we figure out where that cell is and that cell and that, and only target those, just like that first slide, then we can get information on just the cells and we can ignore all the other things. If we do that, this is how it really works. Don't go into the slide much. We image the side, we use some software to find them, we figure out where it is, we do a slight array around each one, we target those, and then we're able to do this. All this is automated. We feed the locations into the different mass spectrometers, but this is how the approach works. It's really a sampling protocol, and then it works amazingly well. We have undergrads that come into the group and they do single cell measurements after about a month or two of training because everything sort of is now robust. The hard part of this is getting the cells out. This is just showing you rat pituitary cells. Each one of these, it's really hard to see. There's actually about 10,000 individual dots here. We made the cells visible by using a nuclear stain. We then use software to say if cells are too close, we don't hit them with the laser, but otherwise we targeted about 7,000 cells here. And every one of those, we get a spectra of the peptides present. We can do lipids too, or other molecules. And then we can do things like principal component analysis in this mess and say, here's a spectra from that cell or that cell. And we can get data from literally thousands of cells. I think we're one of the only groups doing this approach, but it would work with anybody who has this type of mass spectrometer. Let me give you an example that most people intrinsically understand a little bit easier. This is showing you an islet of Langerhans. These are the little microorgans that have beta cells that make and release insulin that controls blood glucose, and hence it's misregulated in diabetes, whether it's type 1 or type 2. And they have very peptidergic cells. And so we can take a rat islet, we can disperse it, we can do single cell measurements, we can do principal component analysis, and with no analyte pre-selection, the system automatically separates the cells into those that contain insulin, the beta cells, glucagon, pancreatic hormone, and others. And we get data in terms of the number of cell types, alpha, beta, gamma, delta, depending on peptides, that sort of match literature values. And it changes if you're in the ventral or dorsal port of an islet. And you even can start to see completely unexpected cell heterogeneity in an islet. The pancreatic hormone cells, those that have pancreatic hormone, that's this gene sequence here, single letter abbreviations, methionine, alanine, valine. I apologize if you don't see that, but this is the sequence of it. This part of it cleaved right there from the signal peptide to this GKR site makes pancreatic hormone. And if we were to plot that peak right here, that is these cells right here, each dot is a cell. There's a cleavage here at a double basic site, and that makes a smaller peptide. And we see that here, basically, it's not cleaved except in a few cells. And that cleavage produces a peptide that people expected would be present. And then later on by measurements was said it's too low a level to be biologically active. But it was already characterized that this peptide hits a neuropeptide Y4 receptor and potentiates, changes insulin secretion. Then when people measured it, they said, oh, it's not there. Well, the answer is it's not there except in a few cells. And we reproducibly find a few cells, presumably that have another processing enzyme to make this peptide, but only in a few cells. So that's why you want to do single cell measurements, is you want to be able to understand what happens to individual cells that would normally be completely overwhelmed if you measured all of the cells, all the pancreatic hormone cells. Let me give you one more example of analytical chemistry, and then I'm going to go right back to islets. This is the lipid mass range from a cell right there. And after we've done that, if we say, wow, that cell is interesting, I can carry it across the street to my other lab at the University of Illinois. I have two labs separated by actually about an 8-minute walk. and find the same cell, never missing it because we're good at targeting a laser. And this is now on a higher resolution instrument. This was a couple $100,000. This is actually over $1 million. It performs better. Now we get much narrower peaks, and we can see if we need to, it's also slower to do, we could measure what's in the same cell with a better instrument. And here it is using SIMS and MALDI. So different flavors. We can keep on measuring the same cell because we're only using about 10% of the material in a cell. We've made a device where we can say, go to any particular cell. We can do this for mass spec imaging and extract the cell and do Kaplan electrophoresis mass spectrometry. This is a metabolite profile of a cell after we did peptide profiling with MALDI. And I showed you that because now I'm going to go back to the islets. This is the glucagon mass peak. So these are alpha cells. There's no insulin or pancreatic hormone. And these are the amino acids present in there. And I can actually say, okay, do the alpha cells containing glucagon also have dopamine? And there's this particular cell. I know it's a glucagon cell. I know it's dopamine. I even can spike dopamine or do tandem MS. And so this was the first example of the proof that dopamine is in alpha cells. When we published this type of data, I was contacted by the American Diabetes Association and said, can you do this on human cells? We really would like you to. So they're now funding my group, and we're getting transplant quality human islets. They're isolated from the University of Pennsylvania at a transplant center. And here they are. They're still living. They will isolate from an accident victim, 1 million islets. They will transplant them into type 1 diabetes that have no islets, and they'll send us a few hundred of them. And so these are transplant quality, but we're not preventing a person from getting a transplant. These are individual cells isolated, and we can do the same thing. We can see insulin, now human cells, somatostatin, glucagon, and we can even see data that looks like this. This is from an individual, 4 different islets. Each dot is a single cell. This individual's islet number 4 has a lot of somatostatin. For example, islet 1 had a lot of glucagon, alpha cells. And so we can start looking at this. And now what we're doing is we're looking at this as a function of type 2 diabetes progression. I would probably have more data to show on this. This is unpublished data, but in the last year, some of the islet centers really had to shut down because of COVID. And so we really didn't get many samples, and it's just starting again. And so this is like many of us, research gets delayed because of the last year. If you're not interested in peptides, I've mentioned lipids. Here are rat cells spread out on a target. We're using the higher resolution mass spectrometer. We can see a lot of decimal places because this is a Fourier transform ICR mass spectrometer, and we can see lipid heterogeneity in individual cells. This is something that really can't be looked at in many ways. And what are the cells? I had a student, Elizabeth Newman, who figured out a way to do immunohistochemistry on the cells. To give you an idea, though, she scatters 500,000 cells on a slide, she analyzes just the center part, about 40,000 of them. She then fixes the cells that, so fixing is cross-linking, and you have to do that before immunohistochemistry or immunocytochemistry. The cells stay present, and then only a few cells move. And then we do staining, for example, for a GFAP, something that indicates it's an astrocyte versus neurofilament for a neuron. And now we can say, oh, this cell right here is also positive for an astrocyte, or this one is a neuron. And now we can cluster the cells and see what lipids do we see predominantly in astrocytes or neurons, or are there any? And this is just showing us some data that was just published about a year ago, where we can say these are presumably astrocytes or these are neurons, and we can get profiles of lipids based on cell type. We can now do this, 6 months later on a different mass spectrometer. This is 30,000 cells. So again, high throughput, and we actually can cluster the cells based on lipid profiles. And interestingly enough, even though the mass spectrometer changed, the year changed, the lipid profiles that make them mostly astrocytic or neuron-like seem to be consistent. And one way we can show that is with the higher resolution instrument, we can see things like sphingomyelins. And the sphingomyelins are only coming out in the cells that are neuron-like, as they should. We couldn't measure that before on the other instrument. And so we have the ability to look at thousands of cells from the brain, from any organ, and look at lipid heterogeneity. Let me just show you one ongoing research project. This has not been published. It's actually under review right now. And this is with Arnold Creakston. And Aparna Badura, who's a new assistant professor at UCLA. She was working with UCSF while we did this. And this is 150,000 cells. And what we're looking at is human development. And it's, again, we're dispersing the cells, measuring them. The cells were dispersed at UCSF and sent to us. And what we've done is we've clustered them based on brain region. And this slide is just way too much information, but it's basically showing you particular mass peaks in each cluster. The clusters are distinct. They have reproducibly distinct lipid profiles. And the lipid complexity then we can see by brain region. And we can actually see this during development. And so again, what we're doing now is, linking this to transcriptomic data, but this is type of information about lipid heterogeneity that is not available using other approaches. And it sort of started out with this single cell at a time till I had a graduate student that said, let's make it high throughput. And then we've sort of gotten to the point where we're doing studies with hundreds of thousands of cells. I only have 2 more pieces of data, and let me sort of show you where this is going. This is some, again, unpublished work, but shows you one direction we're going. If the material is left behind, what else can we do from a single cell? Well, the most common measurement for single cell measurements right now is transcriptomics. That is, what is the RNA in a cell? And Jim Eberwein, who we're working with, is actually the person who did the first single cell transcriptomics. And I think he published this in 1991. And he asked, can we do transcriptomics and mass spectrometry? And so we've been working on this, and this is just showing you some results. We have some cells that are left there. We coated it with some matrix. We, in this case, image it, and we find individual cells here. And these are the lipid mass spectra for the lipid region. We can then isolate those. Jim Eberwein at the University of Pennsylvania. If you don't know, that's about it's over 1,000 kilometers away. So this is not adjacent labs. We'll then take these cells that we've isolated after mass spectrometry and do transcriptomics. And here's just another example. He's doing his approach, and he's getting in these 2 particular cells, he's getting quite a few transcripts from individual cells. Now you have to understand, we did transcriptomics completely wrong. We didn't isolate the cell and add an RNA ACE to destroy RNA degradation enzymes. We didn't isolate it using the right solution. We added DHB, a matrix good for mass spectrometry. Then what we did is we dried it, put it into the vacuum system, hit it with a laser. All of this has nothing to do with optimum transcriptomics. And then after we did that and selected the cells, Jim Eberwein did transcriptomics. However, the power of transcriptomics is such as even though the sample preparation was done wrong or done for mass spectrometry first, we still get useful transcriptomic data. Now, I will admit, one of the reasons this hasn't been published is that we find that this works 50, 60, 70% of the time, and 30% of the time, or 40, we get nothing. And we're still trying to understand what happens. Just to tell you, the way it works now, when you have 2 labs that are doing something, my lab wants to say they must have did something wrong, they blame us. And so now we have the scapegoat. The person we think must be doing something wrong is, FedEx or, DHL, the delivery people must, occasionally do something because we can't figure out why it fails a third of the time. But when it works great. And so once we figure out those details, we'll be publishing this. And then the last example is, we're not the only group doing single cell measurements, but can you go smaller? What about what's in a cell? So here are 3 examples from my group looking at this. This is a single dense core vesicle. It's from an aplasia cell. It's a big vesicle, and we can do mass spectrometry on it. This is in an adjacent cell, a red hemodeck vesicle. It doesn't have too many peptides in it. So we can do individual organelles. This is SIMS of a cell looking at vitamin E, and we see vitamin E is concentrated right at the spot where we damaged an axon. And lastly, you can have a cell soma in a process. We added about 100 picoliters of matrix in different locations, and we get spectra, and we get lipids and peptides. So we can say peptides are starting here and going towards a release site. You definitely can go smaller. This is individual isolation. We can make this automatic. These are, again, the dense core vesicles. There's one and there's, we can, this is what the image really looks like. We can stabilize those and then do an image mass to find these locations. And once we have them, we can hit them. If you want to go smaller, then about a half a micron, it becomes hard to target it with optical microscopy. So we've even done scanning electron microscopy to find sparse objects. There is one right there. And once we do that, we know the location. Now we have something to hit with our laser. And even though the laser footprint is bigger than the object, we still hit this and can see the molecules. And when we do that, for those dense core vesicles, we see this uniform population of peptide-containing dense core vesicles. It gives you good spectra. There are some of the lipids. This is the mass region for peptides, pretty high quality spectra. We discovered that based on k-means clustering, we have 3 different populations of vesicles that have slightly different overlapping contents. So these are what they call violin plots, and it's basically the number of cells with each of these masses. And for example, the green cluster over here has mostly these mass peaks, a little bit of some of the others. The red cluster has, for example, these mass peaks. And we know what some of these are. These are peptide-like molecules that are pheromones. This is an exocrine gland. And we're able then to look at heterogeneity for objects that are quite small. Mass spectrometry has the raw sensitivity, raw detectability to make this work. And what really requires is sample handling to prevent dilution. And that's what I've been mostly showing. We are just adapting these types of measurements, for example, to work with mammalian cells, but it really does take a lot of sample preparation, because if the volume of a dense core vesicle is a femtoliter, what we're trying to do is prevent it from spreading and diluting, for example, to a microliter, because then we wouldn't be able to detect it. Okay, I'm done, and I just want to end, with a couple conclusions. The technology is there to allow single cell measurements to work. Mass spectrometry has the detectability to do this in a non-targeted manner. And when we do that, in every cell that we've looked at, in every animal that we've looked at, cell heterogeneity is higher than most people really expect. It is not, when you look at an average and you say, oh, for example, this amino acid or this transmitter is present at this level in the brain, the answer is it's actually a lot of cells are 0 and a lot of cells could be millimolar. And then the average might be low micromolar. So we're finding lots of unusual peptides, lots of small molecules or lipids that aren't expected because of our new tool to look at this range. And this is a really fun and exciting area that is opening up a lot of understanding of health. I didn't go into it, but then this regulation then tells us something about disease. And so, but to be honest, if you have a new tool and you're trying to understand how it works, one of the first things I like to understand is how well it works under normal circumstances before we start looking at, diseases. And that's what I tried to emphasize today.